we will go back again towards the west and towards Portugal, where we have node and user presentation with Ângela Inácio, University of Coimbra again, and Luisa Cortes from the Portuguese platform of PPBI, presenting study titled Aberrant Hippocampal Transmission and Behavior in Mice with a stargazin mutation linked to intellectual disability. Ângela, over to you.
Great, thank you. So good afternoon, everyone, and I would like to thank the organizers for the opportunity to share my work here. So I'm a postdoc at the Synapse Biology Lab, which is led by Ana Luisa Carvalho at the Center for Neuroscience and Cell Biology in Coimbra. So glutamate, as you all know, it's the major excitatory neurotransmitter in the brain. And the glutamatergic system plays a very important role in neurotransmission and synaptic plasticity modulation during development. So therefore, it's not surprising that mutations in genes that code for synaptic proteins have been associated with increased risk to develop psychiatric disorders. And one such gene is the gene encoding the protein stargazin. So stargazin is a transmembrane AMPA-regulatory protein, which was originally discovered in the stargazer mice. So this mouse is a natural knockout that doesn't express stargazin, and these animals have severe ataxia and also have absent seizures. As I said, stargazin is a transmembrane protein, so it has 4 transmembrane domains, and both an intracellular N-terminal and C-terminal. And the 3rd and 4th transmembrane domains, as well as the extracellular domain, are very important for the physical interaction of stargazin with the glutamate AMPA receptors. So it is well known that stargazin regulates AMPA trafficking to the synapses and also stabilizes AMPA receptors there, as well as it plays a role in regulating the gating of AMPA receptor channels. Also, given its role in the regulation of AMPA receptor trafficking, this protein has been shown also to play an important role in mechanisms of synaptic plasticity, such as long-term potentiation, long-term depression, and also homeostatic plasticity. And stargazin belongs to a family of proteins that have complementary expression patterns in the brain. And in the case of stargazin, it's highly expressed in the cerebellum, but is also present in the cortex and in the hippocampus. So in 2011, a new missense point mutation in the gene encoding for stargazin was identified in a patient with intellectual disability. And this mutation leads to the replacement of valine for a leucine at position 143. Which is located in the 3rd transmembrane domain of the protein that is important for the physical interaction of stargazin with AMPA receptors. And we know that this residue is highly conserved among species, and using bioinformatic tools, our predictions were that this mutation might be very damaging for the protein function. So in order to understand how this mutation would affect stargazin function, what we first did was to transfect cortical culture neurons, either with the wild-type form of stargazin or with the mutated form associated with intellectual disability. And we image stargazin diffusion by using quantum dots. And what we observe is that when compared to the wild-type form of the protein, the mutated form of stargazin has increased mobility in synapses. It has a decreased residence time and an overall increased diffusion coefficient. So At this point, we were convinced that the mutation affected the function of the protein, so we decided to move forward. And in order to understand how this mutation might impact circuit formation in vivo, we decided to generate a knock-in mice expressing the mutation, the human mutation identified in the intellectual disability patient. And for that, we used an homologous recombination strategy. And what we first did was to look at the morphology of the brains of those animals. And as you can see here, there are no gross alterations in the morphology of the animal, of the brain of the animals carrying the mutation. And we also looked at the expression pattern of stargazin. And as you can observe here, there are no major alterations in the regions where the protein is expressed, as well as in the total levels of expression of stargazin. So what we did next was to perform a set of cognitive behavioral tests in order to see if the animals presented any alteration, behavioral alterations that might correlate with the symptoms that the patients show. And just to show you one of the tests that we performed. So we did the object displacement test, and in this test, in the first day, the animals are allowed to explore two objects in an arena. And 24 hours later, the animal is put back on the arena, but one of the objects is displaced. And usually, if the animal remembers the previous location of the object, what it happens is that it's explored for longer periods of time the object that was displaced. And in fact, this is what happened in the case of wild-type animals. So they explore above chance level the object that was displaced, whereas in the case of the animals with the mutation, this does not happen, which suggests that these animals have impaired cognition. And since alterations in social interactions are also a very common feature in patients with neurodevelopment disorders, we also assess social interaction by performing the three-chamber test. And in this test, the animals are allowed to explore a cage with an animal inside or an empty cage. And since mice are very social animals, usually they tend to spend more time interacting with the animal instead of exploring the empty cage. And this was true for all the animals, the wild types and both the heterozygous and homozygous animals carrying the ID-associated mutation. However, when in the second part of the test, we look into alterations in the preference for social novelty. So in this case, what we do is that instead of having an empty cage here, we place a new animal. And whereas wild type and heterozygous animals spend more time interacting with the new animal, the homozygous animals fail to do so, which suggests that they have either impairments in social memory or in preference for social novelty. So to better understand what were the mechanisms behind these behavioral alterations and whether the mutation might affect glutamatergic signaling in these animals, we started by recording mini excitatory postsynaptic currents mediated by AMPA receptors in CA1 hippocampal neurons. And contrary to what we were expecting, given the role of stargazing in the trafficking of AMPA receptors, we didn't observe any differences in the amplitude of these events. However, we do observe a significant decrease in the frequency of the events. And usually a decrease in the frequency of mini excitatory postsynaptic currents might be either related to changes in presynaptic function, for example, changes in the probability of neurotransmitters release, or it might be related with a decrease in number of active synapses. So to better clarify this, what we decided to do next was to perform some field recordings. And for this, what we did was to stimulate the shuffle of collaterals and perform field recordings in the apical dendrites of CA1 apical neurons. And when we stimulated the slices with stimulus with increased intensity and evaluated the amplitude of the fiber volley, which gives us a measure of the presynaptic activity, we don't observe any changes in between genotypes. And the same happens when we look at the field slope of the excitatory postsynaptic potential, which gives us an idea of the postsynaptic function. And we also, I'm not showing it here, but we also did some paired pulse facilitation experiments, and we also didn't observe any changes in the probability of neurotransmitted release. Also using this same type of strategy, and since stargazing plays an important role in synaptic plasticity, we also induced LTP in these slices using a theta burst protocol. And as you can observe here, there are also no differences in between genotypes. So at this point, we were a bit puzzled with the results because our decrease in the frequency of mini excitatory postsynaptic currents suggested that we had either an alteration in presynaptic function or a decrease in the number of active synapses. But when we recorded when we performed field recordings, we didn't observe any differences. So here's when microscopy gave us a great help in deciding how to move forward with the project. So we went back and looked into the expression pattern of stargazing in the hippocampus. And as you can see here, when we compare the expression of levels of stargazing in apical dendrites, where we were recording from, with the amount of protein that is expressed in basal dendrites, we see that stargazing is much more, the intensity of the staining is higher in basal dendrites. So at this point, what we thought is that maybe the alterations that we were observing in the frequency were a result of alterations specifically in synapses in basal dendrites, but not in apical dendrites. So to test this, what we did afterwards was to perform again field recording, but this time we stimulated and recorded from basal dendrites. And again, when we look at the presynaptic function, we don't see any changes. However, in this case, when we look at the excitatory postsynaptic potentials, we see that there is a reduction in the animals, in the slices of the animals carrying the mutation compared to the wild type. And again, we also performed a theta burst protocol to induce LTP in the slices, and we see that although we still can induce long-term potentiation, it is to a smaller extent than what happens in the wild type. So it means that in this case, we have impaired LTP in synapses in basal dendrites. So our next step was to try to explore this further and try to understand what was happening. So we decided to look into the morphology of the neurons in the CA1 region. And for this, what we did was to inject virus in the tail vein of the animals to induce GFP expression under the promoter of synapsin. And in this way, we could achieve a sparse labeling of neurons in the brain. And afterwards, what we did was to perform some confocal microscopy, and we did a 3D reconstruction of the neurons using the IMARI software. And when we looked at the complexity of those cells by performing shoal analysis, we didn't find any significant differences between genotypes. And the same was true when we look at the total dendritic length, if we look into basal or apical dendrites. And taking advantage of this same strategy, we also decided to look into the density and morphology of spines. And when we look into the total density of spines in basal dendrites, we don't see any changes in density in between genotypes. However, when we look into the density of more mature spines, this is the case of mushroom spines and stubby spines, we do see a significant decrease in the density of this type of spines in the animals that carried the disease-associated mutation in the stargazin gene. And this was accompanied by a significant increase in in the density of more immature spines, here both syn and philopodia. And as our electrophysiology results suggested, when we do the same type of analysis, but looking into spines in apical dendrites, we don't see any differences. So lastly, what we did was to look into the ultrastructure of synapses by electron microscopy. And what we observe is that when we compare the PSTs of all type animals with the PSTs of animals carrying the mutation, we see that there is a significant decrease in the length of the PSTs, which is in agreement with what we see when we look at the spines. So we have a decrease of mature spines, which have larger heads, and we see an increase in the number of immature spines, which have smaller heads. So these results are in agreement. So as a conclusion, although I didn't show you all our data, our in vivo experiment showed that stargazing mobility is altered, and also that the ID-associated variant of stargazing impacts AMPA receptor trafficking. Also, the knocking mice expressing the disease-associated stargazing variant display alterations in social interaction as well as cognitive defects that correlate with the disease phenotypes observed in patients. Moreover, when we looked into the spines in the hippocampus of these animals, we see that the mutant form of stargazing impairs the maturation and glutamatergic synaptic transmission, specifically in basal dendrites, which suggests that apart the well-known brain region differences in the function of type proteins, that might be also more subtle differences that are defined by the intracellular pattern of expression of type proteins. And although I didn't show you this, if you want to look at in the paper that we recently published, we also studied the composition, the biochemical composition of the postsynaptic densities of the animals carrying the mutation, and we found some alterations in the levels of stargazin at PSTs, as well as alterations in stargazin phosphorylation. So as a main conclusion, we can say that our data suggests that this mutation has a causal role in the pathogenesis of intellectual disability, and also our data also shows a new role for stargazing in the maturation and function of hippocampal synapses. And I would like to finish just thanking everyone in the synapse biology lab that contributed to this work, particularly Ana Luisa Carvalho, my supervisor, and Gladys Caldeira, who started the project and also generated the animals, and all our collaborators. And if you have questions, I'll be happy to answer them.
Thanks a lot, and it's fascinating that you had a talk on this receptor and the functions following the one that we saw from Bordeaux. So it would be nice to see those methodologies being combined.
Yes.
How is it, Luisa? Were you going to say something, or?
Yeah, I just would like to present the note if it's possible.
Yeah, please. Go ahead.
Okay, can you see my screen?
Yes.
Okay, just as a brief overview of the PPBI. So it's the Portuguese platform of bioimaging, and it's basically a multi-site research infrastructure that is a combination of 17 sites distributed along Portugal. Portugal is not so big, so it's quite easy to go from one to the other. And Ângela just presented the work done in Coimbra, in the Center for Neuroscience and Cell Biology. And it's also very close by of the other facility that just presented early this afternoon, the BIN, which is quite nice because then if you come to Coimbra, you can combine different modalities of imaging, just the cellular or more biological imaging and medical imaging. And of course, because we are distributed in different research centers, we also have expertise in different research areas, not only neurosciences, but all the other biological areas. We like to organize stuff, not only trainings for related to microscopy and image analysis, and we are very happy to accept everyone that are interested in participating in our courses, but we also managed to organize meetings on microscopy, and especially the one that is combined with the infrastructure from Spain, that we do it annually, once in Portugal, once in Spain. And we are happy if you want to join us as well. All our information is on our website, so you can find information about all the sites, all the notes from PPBI, and also all our activities, and also for imaging analysis, you can also find some resources that we develop in the notes. And of course, we are nothing if we don't have people, and all these faces are what will support you if you came to PPBI and one of the sites. And thank you for your time.